Our technology is based on taking a small tissue biopsy from the stomach of the patient and separating the extracellular matrix and the cells from this tissue, while the extracellular matrix, the biomaterial, is processed into a personalized hydrogel. The cells are reprogrammed to become embryonic stem cell-like cells. And then when we put the cells inside the hydrogel, we can mimic the embryonic development of the spinal cord and develop 3D neuronal networks with the motor neurons.
The process of what we do is we take from the patient itself the omentum, which is fatty tissue from the abdomen. We can separate the cells and the extracellular matrix. The cells are reprogrammed to become iPSCs, stem cells, while the extracellular matrix is processed to become a hydrogel. Then we can mix between the two to generate the implants. We differentiate them into spinal cord motor neurons implants, and then we can implant that into injured spinal cord into the patient.
We hope that the integration process of the cells of the implant will create functional connections with motor neurons or sensory neurons in the spinal cord and will improve functional recovery of the patient.
So based on this technology, we recently formed the company Matrisoft, and in the future we're going to transplant human tissue implants in paralyzed patients, helping them to walk again.